Let me know when I should share my screen again.
Okay, yeah, you're ready to go ahead and share your screen. Okay, there, folks. Um, all right, welcome everyone. It looks like we are people are slowly joining the room. Thank you so much for tuning in. I know things are, for a lot of folks, this is a busy time with end of the semester wrap up things and just holiday fun. So thank you all so much for joining us for our last webinar in the checking in on TCNs and their networks of digitization networks. My name is Jill Goodwin. I'm the conference manager for iDigBio. We first have Erika Tucker with us, who's going to be talking about TPT, and then and then Nelson Rios will follow. We will pause for Q&A in between. So if you have questions during Erika's presentation, please feel free to add those to the Q&A and we will address as soon as we can. You can also alternatively raise your hand and we will call on you again after the presentation. So we are aware that we are seeing some other things in this presentation here. I think it all looks good and we are ready to get started. So Erika, take it away. Thank you so much again for being here.
Yeah, hi everyone. So my name is Erika Tucker. I'm the Community Engagement Projects Manager at the Milwaukee Public Museum, and one of the projects that I'm managing is the Terrestrial Parasite Tracker TCN, along with lead PI Jen Zaspel. For the Terrestrial Parasite Tracker Project, or TPT for short, we are focused on digitizing arthropod parasite collections to trace parasite host associations and predict the spread of vector-borne disease. The project started back in September 1st, 2019, and we've obtained 3 no-cost extensions and hopefully will be finished by mid next year. The TPT project is made up of 24 primary data providers from across the U.S., 11 collaborating institutions, and 7 supporting organizations. This includes 36 project PIs and co-PIs, 13 of which were new to the TCN program back when we first started. The TPT project chose to focus on parasitic arthropod digitization for a few reasons. Many arthropod ectoparasite groups feed on vertebrate hosts. This includes parasites that vector numerous diseases and pathogens. And because of this, parasitic arthropods inflict an enormous burden on the health of their hosts, both directly and as vectors of harmful microorganisms. Parasites represent a substantial proportion of biological diversity, and despite the importance of such parasitic arthropods to human and animal health, and in understanding biodiversity in general, parasite data is underrepresented in the specimen data currently digitized and available. With digitized parasite data, we would be able to better examine biological associations between parasites and hosts, learn more about disease ecology and how pathogens are vectored and spread, use georeferenced specimen data to facilitate the creation of distribution maps for parasitic taxa, both for control measures and conservation management. And of course, digitized data aids in a variety of applications related to alpha taxonomic studies, comprehensive systematic approaches, and trait-based analyses. The goals of the TPT project were to provide baseline information about parasites by transcribing, imaging, and georeferencing at least 1.2 million arthropod parasite records from collections across the U.S., to capture 500,000 parasite host association records, and to publicly share all digitized records and association information with data aggregators such as iDigBio, SCAN, GBIF, and Globi. With the goals of capturing so many records from so many data providers and partners, we had to incorporate multiple methods into our workflows. For record transcriptions, we used 2 primary means of capturing data: in-house transcriptions by staff, students, and volunteers. Depending on the collection, this could be data transcribed directly into a collection management system or database, data transcribed into a spreadsheet for batch uploads to data management systems, or some combination of both of those or something in between. Our second primary way of transcribing records was to harness volunteer power through the crowdsourcing platform Notes from Nature. For this, we created information training guides on the platform for volunteers for each expedition or batch of records from a particular collection. We gave each one a fun name along with details about the collection and the records that we were asking volunteers to help with. And these are just some pictures of our different training guides showing what everything means on this particular slide. Notes from Nature also provided us with a lot of useful statistics that could be used for various reports or to improve how we interacted with our volunteers. And as you can see, we had quite a lot of volunteers through the entire duration of the project. For imaging, we needed to be able to digitize specimens that were in a variety of different mediums, from flat slides to round vials to super fragile pin specimens. Kind of had a little bit of everything. For both our slides and vials, we used flatbed scanners to capture low-resolution images. We created rigs out of either paper or plexiglass, depending on the situation, to consistently place specimens in the same position on the scanner, and made templates in the free software program InSelect that we could then run our specimen scans through to automatically separate individual specimens into individual images, and in many cases, even automatically capture some of the text information on the specimens, like catalog number or other information to help label the images produced. To capture both high-resolution and low-resolution images of our pin specimens, we teamed up with Macroscopic Solutions and obtained a number of macropods for the project. Macropods have the capability of taking high-quality photos of really teeny tiny things. Oops. And yeah, really teeny tiny things with its microscope lens attachments as well as other attachments. And for our project, since they're parasites, many of our things are really teeny tiny, so we needed that. Just as important as the equipment itself was all the tech support that was provided by Macropod Solutions and the many tutorials that are available online. Some of these tutorials are also really useful even if you have a different camera setup. For capturing and sharing parasitic association data, we partnered with Globi, the Global Biotic Interaction Database. Globi is an open access data integration platform that continually indexes existing publicly available species interaction datasets, literature, and specimen records using open source software. There are many different ways that our collections could connect with and share data on Globi. So no matter what collection management system we were using at a particular institution, we could all be accommodated. Once our association data was connected to Globi, it could then be found by others through a simple search on the database from the homepage with this simple search option, or by going to the Browse tab, which also provides the option for downloading multiple association records as a CSV, as well as providing different ways to visualize the data. Pretty quickly into our digitization process, we realized that we needed to work on curating our taxonomies and name lists in order to actually get a lot of what we were capturing properly into our databases. So, we created a number of taxonomic tools and resources that can be found on our TPT Taxonomy Resource Hub. This includes taxonomic name lists for terrestrial parasite groups like fleas, ticks, mites, and lice. These lists have multiple formats available so that you can get them into your particular database and can be used for a variety of purposes like database backbones or research. Excuse me. Taxon-specific contact information for the experts associated with each name list is also available on the site. We also included a bunch of useful R cleaning tools and scripts for creating the name lists that are available on the site, and these can be used to create new name lists as well. Since this is a GitHub-based site, it makes everything easy to update by either our TPT group or the greater community. It can also be versioned when significant changes are made and can have citable DOIs applied. Excuse me, applied as needed. Sorry, I have a little bit of a cold. Since a big part of our project is capturing association data, we also worked with Globee to create a way to search and curate custom taxon association lists. Globee also provides many pre-compiled association datasets that are already ready to download. If I can stop coughing. This includes, of course, many parasite association datasets from the TPT project. You see the list there, just some of them. Now, as you might imagine, with all these moving parts, we also needed a number of different management tools. We used Google Spreadsheets for self-reporting by PIs on general project updates, as well as individualized spreadsheets for institutions to keep track of their digitization numbers. We also created digitization progress reporting graphs. These progress graphs showed not just the project start and end date, but the rate at which a collection is currently digitizing and the rate at which they need to be digitizing right now to meet their goals. These graphs can be made for overall digitization of a collection or a group, or what we often like to do is to make a graph for each type of digitization a collection is working on. One for the filed specimens, one for slide mounted, one for images, transcriptions, etc. And you only have to enter the data once to produce multiple different graphs. The script and detailed instructions on how to use and modify this are all available and free on GitHub. We found that these graphs are super useful tools for collections in figuring out not just if they are on track with their digitization goals, but where and how they may need to tweak things to get back on track if they need to. We've also added all of our workflows and protocols to the bug guide repository, or bug flow repository. Bug guide is a different thing. Fun also, but Bugflow is a community resource for developing and sharing entomological collection digitization workflows, protocols, and modules. By adding our exemplar workflows to Bugflow, we not only make sure that everyone on the team has access, but everyone in the greater collection community has access as well. It's a really nice spot to share current protocols, get new ideas, and just generally help build the collection community resources. There are basic workflow outlines for pretty much every type of digitization entomological collections may run into, including everything from planning for a digitization project to proactive data capture in the field and pre-curation, all the way through imaging and data cleanup. All resources are free to use or modify for your specific collection. And these are just some pictures of what you might find on there for particular modules. There's instructions for the InSelect program I mentioned earlier, and different pictures of different setups and protocols. For help with training folks about association data, we created a reusable lesson plan. Our lesson plan, the Interaction Data Interpretation Workshop, is freely available online. It explores many potential issues that come up when trying to capture interaction data in databases or as part of a digitization protocol or other activity. The lesson plan was built using the Carpentries Workshop model, which has a GitHub backbone. We like to use GitHub. This makes it easy to use the lesson plan as it is or to contribute improvements or updates. There are also detailed instructions available from the carpentries on how to make a copy of the lesson plan and modify it for your own needs, or you can use it as is. And with that, I will give my voice a break and say thank you for listening and take any questions if there's time.
Thank you, Erica. Good job. I feel like yeah, that was yeah, that was a wonderful way to keep uh powering through. I'm so sorry that you're
Oh,
dealing with
no.
that cough.
I apologize for coughing during that.
No, 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 you did great. I feel like that would have been hard for me to keep focus. Anyone, if you have a question, you can put it into the Q&A at the bottom, or feel free to raise your hand and we can unmute you. We can also wait until the end too, if there are any questions, so Erica can have a moment. I'll give folks a second here. Okay, all right. I'm going to have Nelson have you share your screen. Thank you so much, Erica. And if you can stick around just in case folks have questions at the end, that would be great. Thank you so much. Okay, looks good. We are ready when you are. Up next, we have Nelson Rios from the Peabody Museum talking about ESB and DigIn workflows. Thank you so much, Nelson. Take it away.
All right. You hear me fine?
Yep,
All right.
sounds good.
All right. So I'm going to talk about ESB and DigIn TCNs. Oops, not advancing. There we go. So first off, I'll start off by introducing the two TCNs and talk about the similarities between them. So the ESB TCN is a TCN that's focused on digitizing marine mollusks from the eastern seaboard, while the DigIn TCN is focused on non-molluscan marine invertebrates from all around the world. Both projects had very similar start dates around, the fall of 2020, with Eastern Seaboard coming first in September, and then November that same year, DigIn started. Eastern Seaboard has a target of about 4.5 million specimens, and DigIn was about 7.5 million specimens. So as I mentioned, the Eastern Seaboard has a geographic scope, and that actually presents challenges in itself, because when you're trying to digitize data, until the data are digitized, you don't know where they're from in a very easy fashion. You can't just say, oh, I'm going to take everything, look it up in a spreadsheet, because those spreadsheets don't always exist. So it is kind of challenging in determining which taxa are within the range of the geographically delimited area that we're focusing on here. We're defining the Eastern Seaboard as basically the exclusive economic zone around the United States, which is about from shore to 200 nautical miles offshore in most places. And then a little bit inshore to encompass for the places where the estuarine areas and things like that. But we ended up doing a lot more interior stuff because, as I said, defining these areas are difficult. So what often happens is we say, oh, well, let's identify all the marine taxa that occur in this area and then pull all the data for those records and use that for either georeferencing or go in and digitize those records. Well, you do that, you also are bringing in a lot of inshore records when you're doing that as well. And so there has been some, and I'll show this in a later slide when we get to the georeferencing parts, where you see a lot of interior stuff that gets georeferenced. So both projects, you can see a list of the various collaborators here. The ones that I have highlighted in yellow are actually not institutions that are involved in digitization. They're involved in more of providing technology or they're involved. So in the example of Q Aquatics, they are actually doing digitization, but they themselves aren't a provider of data themselves. They don't have a collection or they're not an institution providing specimens. And so we're just facilitating the digitization through technology and access to facilities and resources. So there's three main types of digitization that's happening in these projects. One is de novo transcription, taking brand new specimens off the shelves, or transcribing data off of ledgers and labels. There's also imaging that's going on where, and oftentimes that can be associated with de novo transcription, but not always necessarily the case. And then georeferencing. And georeferencing happens both with new records that are coming in as well as legacy records that are in need of georeferencing. And so, like I said, our role within this project is not, even though we do have mollusk collections and a large marine invertebrate collection, our role is not actually as a digitization partner in the project, but more as a technology support. So I see I misspelled georeferencing there. So, we provided guidance for georeferencing and training, cyber infrastructure support for all the georeferencing technology that's used across the project. And then we also proposed a number of innovations to enhance marine georeferencing and facilitate georeferencing in marine environments. And I'll talk a little bit about those. So most of my talk is going to focus on the georeferencing aspects and the parts that we developed here at the Peabody. So I'll start off by talking about just our general overview of the workflow. And so, we start off with all the data providers at the bottom, both DigIn and Eastern Seaboard, and there is overlap between those two data sets. I will point out two people who you see shown on the slide, VJ Barv at the bottom left and Robin Delpina at the bottom right, where they are instrumental as part of this workflow. They are, for each of those respective projects, they serve as the data managers that facilitate getting data into the georeferencing process and data coming out. So without them shepherding data, this entire process doesn't work as well. And so I think as part of any georeferencing workflow, having someone dedicated to that role of managing the data flows in and out and what's being georeferenced, who's doing what, is very critical. So as part of the georeferencing process, there are institutions that are doing their own internal workflows, whether that's using geolocate inside their own database or using another tool like Google Earth or whatever it is, their process they're using. There are institutions that have their internal workflows for georeferencing, and that happens, and they're doing it their way. The other option that's happening across the majority of the providers within the projects is the data get pushed into a portal like Symbiota, and in this case, it's Invert eBase. And then from there, the data sets, various data sets can be georeferenced directly in geolocate from within Symbiota, and that does happen. And then, they go back from Symbiota back into the parent database. Oops, going wrong direction. But with the most typical process is from that Symbiota database, exports from Symbiota or InvertEBase are made, and they're published into the collaborative georeferencing framework that we've developed. And that's where Robin and VJ both spend a lot of their time, is developing these data sets and defining what is it that needs to be georeferenced and pushing that into the collaborative georeferencing system. Whoops, I keep pressing the wrong button. But it doesn't just have to go in through Symbiota. And what happens a lot with DigIn is institutions are providing direct CSV files, and those CSV files then get uploaded into the collaborative georeferencing. So there's multiple ways of getting the data into the system and back out. And then once it's in the collaborative georeferencing system, we can go back and forth to the geolocate client to have another aspect, is another human involved aspect there, where the humans, now the technicians, have to go through and verify all the automated results of georeferencing and then identify the final result of georeferencing and then ultimately push that back. And so Eastern Seaboard, one of the key differences is that Eastern Seaboard is doing all of its georeferencing in-house. And by in-house, I mean the institutions that are collaborators on the project that are providing data are doing the georeferencing themselves. We're doing it in a collaborative fashion where not each institution is necessarily georeferencing their records, but institutions are defined certain areas. So, one particular institution might say, I'm going to take all of Southern Florida and georeference that area. The other side of the coin is what DigIn is doing, and that is where more of an outsourced georeferencing process. And here we work with Aquatics, which is one of the groups associated with Fishbase, and we provide the data to them, and then they do the georeferencing using the same thing. So it's a smaller number of technicians, but they're dedicated to doing just georeferencing, and that's all they do. And then finally, as the georeferencing is going on, we can output, various reporting metrics and whatnot, and then ultimately repatriate data to collections. And so here we can see this is the current status, or the status of a few weeks ago, for the Eastern Seaboard. And you can see here there are 15 collaborating institutions. Approximately 120,000 specimens have been georeferenced. All of this, like I mentioned, is in-house georeferencing. And this is just what's happened within the collaborative georeferencing data portal. Like I said, there are instances where institutions are doing their own internal georeferencing processes. There are a couple of institutions that are in Specify. Geolocate is embedded within Specify, and they're doing the georeferencing directly within Specify instead of pushing it out to the collaborative georeferencing. And that can facilitate some things, so you don't have to worry about repatriation, but we end up losing some of the rich features of being able to collaborate. And on the bottom left of the slide, you can see just a graph of progress over time. And so what you see are the number of evaluations. An evaluation is basically a technician looking at a result and then either refining that automated georeferencing result and saying this is the final result, or saying it's too ambiguous, we can't do this, we're going to skip this record and pass on it. And then they'll usually have to comment as to why they pass. And so you can see the number of evaluations that happen per month. And I mentioned the project started in 2020, but we didn't actually start georeferencing till late in 2022, and georeferencing really didn't take off until 2024. And that's not atypical for these projects of these size and scale, because without the data, there's an entire process of getting the data digitized to begin with, and then starting the georeferencing. If you start too early in the project, you end up potentially having to go back and re-georeference data over and over again as new data come in, or having some kind of process to try to limit the amount of similar data that matches previous stuff that was georeferenced. So waiting to the end can oftentimes help a lot with georeferencing. And if we look at DigIn, we have a very similar approach. I'm going to go back one so I can just point out, as you can see in this slide, there are a lot of records that come inland, quite a bit. And this is, I think, a lot of it is just due to the challenges of identifying marine taxa. When you say this is a target species and we're going to georeference everything for that, we end up querying much more data than is necessary for that species. In some cases, it could be an entire institution's group as to how that query was made, and it ends up grabbing a much larger slice of data. And so that is a challenging part. But in the end, these are still records that are highly valuable, that are getting georeferenced. So it's still, I don't see it as a bad thing. And then, it's the same kind of picture with DigIn, similar kind of performance. 150,000 specimens have been georeferenced to date, 20 collaborating institutions. But as I said, the digitization, or at least the georeferencing, is happening outsourced and by the group at Aquatics. And once again, we can see that there are a fair number of inshore records that have been selected and provided to this. And it's really just about what the data providers are providing in this case to the project. And the number of evaluations per month are pretty much follow the same pattern, where late 2023, 2024 georeferencing started to take off. And so I'll just quickly go over just a couple of innovations that we've done within the georeferencing cyber infrastructure to help improve georeferencing. And this is all stuff that's been integrated in the Geolocate platform. So we've added a number of new marine gazetteers, and gazetteers are back-end data sets that are used by the georeferencing algorithms to be able to identify, or to help identify, where a particular location may have been collected, a collecting event, where it may be from. And so we mined all the data in FishNet2. We also mined data from MCZBase to create a gazetteer of those previously georeferenced records. We also extracted a number of marine features that were previously not available to geolocate, using undersea features from the NGA GNS server, as well as the International Hydrographic Organization number of world seas database. And then we built a gazetteer of marine cruise ships. And so what we did there is we actually harvested a number of basically research vessel names by mining data from things like Wikipedia and other places where they have references to research vessels. And then writing a number of scripts that were able to mine all these various data sources to start extracting and deriving data sets. And you can see here I have a couple of names of some of the major vessels that we've, in this case, the Anton Bruun or the Oregon Bolero cruises. Now, this data has yet to be integrated directly within geolocate, and part of that is we still have to, derive the algorithms that will parse the locality descriptions a little bit differently, since the data are much different than a typical locality description. And so we're still working on some of that to see if we can make that available. Unfortunately, the project is running to an end, so I don't know if it'll be available for use in this project, but we do have the databases and we've made those available to various georeferencers within the project so that they have access to the gazetteers for georeferencing. We've added a number of marine-centric layers that can now be visualized within geolocate, the exclusive economic zone, the NOAA navigation charts, and a number of different bathymetric layers. One of the things we've also developed is we didn't want to just be able to showcase bathymetry within geolocate. We wanted to be able to actually query it. And so that means anytime you drag a point somewhere on the map, we're able to extract out the depth information of where that point is. But we decided why not do it for elevation as well. The problem with that is that it starts to become very expensive if we're going to host all this data. Right now, we have about 3 terabytes of bathymetric and topographic data that we use for querying. And if we do that using traditional, server resources, it would up our costs about $3,500 a year just in maintaining the raw storage for this. Instead, we devised a new system that allows us to query data sets that are live on the web using normal using cloud-based storage, things like Amazon S3. In this case, we use a system called Wasabi, and it lowered that potential cost from $3,500 to about $180 a year. So some of our development practices are really designed about how we can maintain sustainable practices that will allow us to maintain these services for a very long time without significant cost and long-term maintenance. Another feature we developed is this ability to annotate records in a much more complex fashion than typical. Typical georeferencing is focused on, I have a point and I want to create an uncertainty around it. You may create one little polygon or something to help define it a little better. But with marine locations, we figured things would be much more complicated. We might want to outline an island or identify a number of different points along a start and stop coordinates for a trawl collection or something like that. So we wanted to have that kind of flexibility. So we added all this features, this concept of being able to represent complex geospatial representations and be able to import that into or be able to generate those within geolocate. And so the question is that being used? And I checked this morning just to be certain that people are actually using it. And this was a record that was georeferenced, within the past month. And as part of the ESB project, and it says St. Petersburg Bridge at Boga Ciega Bay, United States, Florida. And the person who did the georeferencing, he added a comment that said polygon made for each bridge going over Boca Ciega Bay. And because he knew that the collection was made at a bridge, but did not know which bridge it was. He said, well, we'll just select all of them, and created little polygons around every single bridge, and then created one, decided to put the point along, one of the bridges that was the most reasonable consensus point, or the center bridge, whichever one, and then create an uncertainty radius that encompasses the entire thing. So this is, so it is being used. Only a small percentage of records are being georeferenced with this level of, additional detail like this, but it is happening. And then finally, the other thing that we developed as part of this project, and this was kind of one of the DigIn developments, is this idea that, data that's coming out of the collaborative georeferencing, or it could be out of any tool, right? Georeferencing is happening. Some third-party tool is being used to georeference from your database, and you have to get it back in. And one of the complexities for a long time has been, well, how do we get data back into Specify? If data get pushed into geolocate, and we spit out this spreadsheet, how do we get it back in? So we worked with the Specify developers, and we also worked with people at Symbiota to ensure that the proper record identifiers were being transmitted properly so that Specify can then create this locality update tool. And as of July 1st, 2024, there's now a function within Specify so that you can take a spreadsheet of georeferenced data if that spreadsheet contains the proper identifiers and use that to update the records within your database. And then finally, I'll just go through, and I would just like to thank all the collaborators. This is a project, as I mentioned before, it's a people-based project. Even though I presented a lot of technology, the work that's being done in this project could not be done without all the collaborators, all the georeferencing and digitization technicians and imaging technicians. So just make sure I want to give credit to all of them. And this is only a small percentage of the people I think that have been involved in this project. So, and with that, I will take any questions.
Thank you, Nelson.
Sure.
That was a lot of new information for me. Yeah, that was super cool stuff. Thank you. I see a lot of, well, I was going to say friendly faces, but friendly names in the audience. Does anyone have any questions or?
Having worked with Specify before, I just want to say that's awesome that you can get data back into the system. That's huge. Congratulations.
Only locality data though, right? So there's
No,
other digitization types, right? So, but that is a big one.
true. But yeah, that's a big step in the right direction. That's awesome.
Well, I'm not seeing any hands raised. I don't see any questions. I think you must have just blown the audience away, folks. Well, I thank you both so much for that. I'll keep an eye out for a second. I would like to give a quick plug again to BioDigicon in September of next year. Let me put this here in the chat. The website is currently it's it is up and running. We are hoping to open up registration in January, and we'll be putting out some calls for sessions too. But this is going to be our largest event to date in Washington, D.C. in September 2025, and we hope everyone can be there. I think, yeah, both of these projects, it would be wonderful to be able, and I guess efforts, it would be wonderful to be able to feature in person next year. But yeah, hope everyone can join us next year for BioDigicon in September. All of the recordings for all the webinars that have happened within this series will be posted if they're not already on the iDigBio page, and can be available for later viewing. So, thank you. Hey, Petra. Yeah, this is all great stuff. Well, thank you all very much. And yeah, hope to see everybody in person soon. Have a great, happy holidays, happy new year, all the things, everyone. Take care. Bye.
Thank you. Bye.